Welcome to All Things AI. My name is Ishan, and in the seventh episode of the All Things AI podcast, we have Dr. Andrew Janowczyk, an assistant professor at Emory University, whose work is primarily centered around developing effective AI to facilitate cancer research. Welcome to the channel, and it's an honor to have you here today.
Thank you for having me. I'm excited to have our conversation.
I'll just start off by asking, how's your day going so far?
Yeah, it's a pretty usual day. Wake up and start grinding through some emails, catching up on the various projects that we're working on, and pushing forward data requests and things like that. So, I think all in all, pretty normal, standard day so far.
So with that being said, I just want to jump straight into the first portion of today's interview, which I want to dedicate to the integration of AI into digital pathology, because I've been following your work for a little while now, and I saw that some of it's dedicated towards, digital pathology and like biopsy scans. So as far as cancer detection, I know that if you do like a biopsy on a patient and then analyze the tissue through pathology, which can be done through the work of a pathologist, then you can see if they have cancer. So I guess on that note, I want to focus in and see, especially in pathology, what is something in recent times that you think is a groundbreaking discovery that's basically facilitated through pathology and cancer detection based off of AI?
Yeah, so I think that's a really interesting question because, and it's highly related to your podcast. I think the largest thing that we've had, and I think you'll probably notice, I don't really use the word AI very frequently. I think internally we tend to be more precise with our terminology. And
Yeah.
I remember this very famous tweet that I saw that other people have now repurposed, and it basically says, if it's in Python, it's probably machine learning, and if it's in PowerPoint, it's probably AI. And you kind of see like the separation of the two groups of people, those that report on it or in the media, and those that actually try and perform this sort of thing. But this is connected to your question, because I think the biggest thing that we've seen is actually the deployment or ability to use deep learning to solve a lot of the problems that we would typically solve. So as you mentioned, my specialization is in computational pathology, where we look at pieces of tissue that are digitized, right? So they're glass slides that pathologists would normally look at under a microscope, and they're now digitally scanned, and we look at them on a computer. This means that it's now basically an image, and then we can process it like computer vision type tasks. So before the advent of, let's say, deep learning, we would use what I would consider more classical image analysis type techniques. And for example, we often want to segment the tumor cells, right? So someone would go and try and build an algorithm to segment the tumor cells, and they would use things like shape contours or try and understand different properties of the cells that presented the images. Like maybe we'll just do some edge detection, and then we can do some enhancements, and then we can look for some circles, but we know we're only looking for circles of specific sizes. And it became a really involved, long, painful process. And one of the greatest challenges there was that when we got a new piece of data from a new organ or from a new tissue site or even from a new collaborator from a different institution, that same algorithm itself wouldn't really work well on that new data because their scanner was a little bit different. So maybe the tissue wasn't as thick. So we had to then go and spend a significant amount of time kind of adjusting these parameters. So really what I'm hinting at is before deep learning, something like a nuclear segmentation algorithm was genuinely a PhD level type thesis. It would take multiple years to develop, and you would develop a deep expertise in the field of detecting cancer cells. Now, with the advent of deep learning, we can go and annotate a bunch of cells and basically provide that to a supervised model and say, this is exactly what I'm looking for. See if you could find this pattern on my behalf. And that same multi-year project that was kind of brittle and very difficult to deploy, we can now actually have it be done in a couple of days. And it'll, so we go from this multi-year process to, a couple of hours or a couple of days, depending on your expertise and the availability of the data set. But that's, I think, really what deep learning is showing us, the ability to take a lot of the tasks that were previously very, very challenging, and now to turn them into data-driven tasks, where we can reduce the amount of time it takes to build these algorithms by literally orders of magnitude. And that creates an opportunity for us to not only do more work, but to look at things in a more sophisticated fashion than before, because a lot of the things that we want to do are not easy to kind of frame in an image analysis type approach. But by just simply labeling data and saying, find me more stuff that looks like this, it becomes really possible to look at things that were hard before.
And so I guess you were kind of hinting at like challenges earlier that you might necessarily have like faced in this process. So I guess relating to that, I want to ask, what are the biggest challenges with the integration of AI into segmentation and pathology? Maybe even something that you faced in your research throughout the years.
So one of the main challenges, and this isn't just a challenge for us in pathology, it's a challenge for all areas, but I think it's um, or all areas of machine learning and deep learning, but in the medical field, it's a little bit more painful. We have challenges associated with domain shift. So if you go and, for example, take the Cancer Genome Atlas, they have about 10,000 whole slide images, and you download those images. Those images come from many different laboratories in the United States. I think it's maybe 30 or 40 of them. When you look at an image, and let's say it's a piece of breast cancer, you look at an image of breast cancer from one site versus another site, they fundamentally look different. Even though the disease is essentially the same, right? It still has the same recognizable features, which is why a pathologist can look at it and fairly trivially diagnose it, say, well, this is obviously breast cancer. But our algorithms aren't currently smart enough to be able to understand what is biologically different versus what is technically different. And technically different in this case can be coming from a different scanner, coming from a different laboratory where they cut the tissue a little bit thicker, a little bit thinner. If you stain the tissue for an extra time or understain it, then it changes the intensity of the images or the pixel values that are in it. There's tons of these kind of compounding factors even before you start to talk about the biology of that specific patient. And the challenge there is that this is why I really don't use the word intelligence here, because these algorithms don't fundamentally understand, okay, this is a cell. Instead, it understands, I'm looking for patterns that have this kind of blue next to this kind of red, and they should be in a kind of a circular shape. And when it sees that, it's happy to light it up for you. But when you transition to a different site and that blue is just a little bit different of a color and that red's a little bit of a different color, those features are likely not going to work as well. So we end up spending a lot of time trying to compensate for this. There's some really good techniques for it. You can use augmentation during training of deep learning to kind of fill in the space of reasonable possible examples that you'll see in the wild. As well, you can do things like stain normalization and stain transfer to kind of try and homogenize it. There's a lot of, that really improves the performance of these and makes them more robust, but it's not perfect. And it's still, to this day, I think, hard to figure out in what cases this is going to fail. So we also don't kind of have an error bounds to say, if this particular slide, you probably shouldn't trust the results, but this other one, you really should trust the results. There's research now that's trying to figure out how to do this more at scale and in an automated, more quantitative sort of way. But I think it's fairly nascent and it's not really ready for prime time. So this is why one of the things that when people try and deploy their algorithms in clinical settings, there's a very long validation process on that particular site, just because you don't know what you don't know. So you just have to keep trying the examples and say, okay, it's really working quite well in these situations. We should start to feel comfortable with it. But there is as well something that's, I think, very infrequently spoken about, a constant monitoring process that needs to take place as well. So we need to make sure that, our algorithm that we validated on day 1,000 is still performing the same way. Not because the algorithm itself is going to change, right? That's a fixed static piece of code with fixed weights. But the actual data that's coming into that algorithm may change because maybe the histotechnician was replaced or went on maternity leave or whatever it is, or maybe the scanner, there's a new scanner, or maybe there's a different microtome, or the microtome over time loses its calibration, so the slides become slightly thicker or slightly thinner, right? So these types of things are somehow part of this quality control process that as well isn't fully in place yet. That's something that as well we're looking into. So it's really about trying to figure out the total scope of this. One other interesting point I would say is that when looking at diseases, what we're trying to do is understand more the long tail. And these are the challenges that I think pathologists will typically face. So obvious breast cancer is obvious, right? There's, we'll just say 80% of disease is really obvious to diagnose, to the point where you can probably get someone from elementary school and say, this is a good case and this is a bad case. And in fact, as an example, not to sound like I'm completely crazy, there was a paper published where they actually trained pigeons to diagnose breast cancer versus normal. And they basically just fed pigeons food every time they got the answer right, and they had little iPads set up with a little button for them to click. And when they clicked the right button, they got fed. And over time, they just learned how to identify breast cancer cases. But I point out that these were easy breast cancer cases, right? They're the ones that you probably, if you were going to be a pathologist, you're introduced in your first year of pathology to kind of get a feeling for it. At the same time, those are most of the cases that we end up having in our training set because they're also the most common. Now, the question is, how's your algorithm, be it deep learning, machine learning, or even your pathologist, going to perform on a case that's rare, that's 1 in 1,000, that's 1 in 10,000, that's 1 in a million? Right. This is also why the idea of using these algorithms for rare diseases is challenging, because it's hard to get enough representative data to actually be able to make suitable inferences from. So in our context, especially in the medical field, getting suitable cohorts is challenging. Well, if you wanted to train, a dog versus cat classifier, you could go to Google Images, say, give me 20 million pictures of dogs, and you get those, right? That's not a problem. But if I'm looking for estrogen receptor positive breast cancers that are between the ages of 30 and 45 that have these specific traits and this mutational marker, now you're looking at a very, very small subpopulation. So trying to find those and curate those cohorts is quite challenging.
I think the thing that I usually hear, I guess, when I talk to like AI researchers on usually is like the amount of data, but now this is opening me up to like, I guess, a new spectrum, because considering that just the type of data that you have can also be a major problem, I think is also important to note. But I guess you're also touching on augmentation. And I know like in my own research, for example, like for high school, obviously it's a totally different thing, but I was trying to use or implement augmentation for the ECG because it's time series based data. So I was wondering in pathology how augmentation works. Is it like changing up the image, like the axes of orientation, or how would that necessarily work?
So we typically take the actual image. It's a 2D image, so it's in basically a RGB matrix. So you have like rows, columns, and then RGB 3 channels behind it. And then we apply augmentations to it. And there's a number of different augmentations. We try and mirror the things that we would realistically see coming from the laboratory. So one of the things that we'll do is, for example, adjust the red, green, and blue values by distorting them. So we'll apply like hue saturation type jitter to kind of just shift them a little bit. You can also go in, we know that the scanners themselves, the microchip that performs the scanning is not perfect. And there's a noise profile that's associated with it. It's kind of a Gaussian noise. So we can as well provide random Gaussian noise or speckle noise into the image to further try and replicate the types of noise that we'll see from those sensors. So I think it's really about understanding where the potential variability, this is kind of the big secret behind augmentation, is that if you look at, for example, PyTorch and Augmentation, Torch Vision, these libraries, they have hundreds of augmentations that you can deploy now, right? And it's easy to turn them on. That's kind of the challenge, right? It's basically half a line of code to say, oh yeah, add blur to this, add this, and this. And the challenge is that a lot of those are not actually going to improve your generalizability, because those augmentations are irrelevant to the actual variability that you're going to see in your data. So the real thing that we try and understand deeply, that we see the most improvement, 15, 20% performance improvement, is to really look at that experimental pipeline and what happens to your data before you receive it, and look at all the possible variability points within it. Try and figure out if there's a big variability or if there's a small variability. Once you understand these kind of things, you look at the big ticket items and say stain variability in pathology is huge. So we spend a lot of time trying to figure that out. But then once we know what that variability is, and maybe we look at pathology images from 5 or 6 different sites and say, oh, the blue range can vary from 80 to 120 as a typical range, with maybe 100 as the mean value. Now that we have that information, we can apply augmentation to our images that fit in that range. Because we know, for example, we're never going to see a routine chemotoxin and ASN image that has the color green in it. It just doesn't exist. It's not a biological thing. Chemotoxin stains blue, ASN stains pink, right? So it's got to be a blue-pink type problem. It makes no sense to go and have an augmentation that introduces green into the image, because now you're essentially training the model to learn that green is irrelevant to the problem, but it's wasting its time or wasting its computation power or parameter space, trying to learn augmentations that are irrelevant. So I think really nailing down and directly mirroring the types of things that you expect to see when you deploy that algorithm, and seeing if you can synthetically generate equivalents or surrogates of those fast enough so we can use for real-time augmentation is really the key to building these better, more robust models. But it comes from a deep understanding of the domain space. It's not something that, if you turn on all of them, you're going to get better performance. In fact, you get worse performance.
I guess the last question I want to ask in this category is, what are the ethical and regulatory considerations with implementing AI into pathology? Because I think, like, from my experience in, like, I guess, interning at a lab, when you get the data, often it's like, or at least in my scenario, it's like already checked for everything and like the patient names are removed. But is there anything else necessarily that's especially concerning in such a process?
So I think we're very lucky in pathology. So I've been involved with a number of different studies, and I kind of see the difference of the requirements for each of them. Digital pathology, kind of current thinking in the world, is that the image itself is already anonymized, right? So in the sense that if I look at this, you have to, of course, remove the patient label, and you have to make sure there's no identifying information about the patient on the physical image. But a piece of tissue is not identifiable. So if I see a piece of tissue, I can't go back and then identify the person that came from, right? Without extensive effort, right? I'd have to like search for that image in a database that also has the patient name, and then do some. It's like it's impossible, if not extremely hard. And that's quite different than if you do, let's say, genomic studies where you're taking the genome of a particular patient, and now you have their mutational profile markers. You can now really directly identify that person very, very easily, right? That's a piece of known identifying information. There's as well some research in the imaging space that's showing that when you take images, for example, of a brain CT, you can actually reconstruct the person's face, and that's super identifiable. So then we need to think about secondary ways of trying to anonymize that data, or make sure that it's used ethically and appropriately to prevent that patient's identity from becoming visible. Digital pathology, once we remove the slide labels and remove all that sort of stuff, we have to look at the metadata. There's oftentimes, not just on the slide label itself, but in the header of the particular file, you'll have maybe the patient name or ID number or some other identifying piece of information, date, time that it was scanned, things that are kind of important for traceability and auditing, but would be identifiable. We can remove all of those computationally fairly easily. So I think that creates an opportunity where in digital pathology, we've been able to, first of all, release public data sets with greater ease. We've been able to share our data sets across different sites in a lot easier of a fashion. And that's really a benefit, I think, for this type of research, specifically because there's some hospitals in the world that happen to collaborate with one of them. It's called the University of Geneva Hospitals. And they have an actual digital primary diagnostic workflow where they scan 1,000 slides a day. And all the pathologists only look at digital images on their computer screen. No one uses glass slides anymore. And this naturally, if you think about it, is creating the cohorts that we need. So now we can go into that clinical pipeline and say, these are the particular patients of interest. We can subject them to anonymization pipeline for which we have ethics approval. And then we now have our research type cohorts. And I think that's a huge game change in computational pathology, because that's not the case for, let's say, genomics or methylation type research, where you need to go and specifically find patients and create research cohorts and then actually do the sequencing and these sort of things. So we have an opportunity to build large scale, clinically relevant, anonymized data sets for research with greater ease once these types of infrastructures are set up.
So with that being said, I want to move on to the second portion of today's interview. And I guess it's really a great transition, because you were just talking about how you're collaborating with the hospital. So that's mainly like what this is going to be focused around, I guess, in a way. So the second part is going to be dedicated to the expansion of AI into healthcare. And so I was wondering, how can the interdisciplinary collaboration between AI experts and medical researchers be facilitated to maximize the potential of AI in healthcare, as far as maybe like clinical trials, or like what can a hospital do, I guess, in a way to aid AI researchers?
So I think there's kind of two points that I want to make. I would suggest breaking down the concept of AI researcher into a specific niche. So I think one of the things that I tell my students, or there's a lot of universities now that offer degrees in data science, and those students come out and they become data scientists by kind of definition, or maybe you want to call them AI researchers. I would argue that AI is kind of a blanket, and there's a lot of other stuff that goes around. And I think the challenge is that an AI researcher or data scientist by themselves are really not very effective people, simply because the real value is data science is kind of a hammer. And the question is how can you use that hammer? Well, I can build a house or I can build a boat, but those are for sure different pieces of information that are required, even if I've mastered those particular tools. So what we end up seeing, and I happen to be an expert in digital pathology and this sort of stuff. I have to be an expert in deep learning, but I would say deep learning within the context of digital pathology is my ultimate expertise. If I wanted to go and, let's say, work on self-driving cars, or even if I wanted to transition to something like radiology, which is still in a medical field, still a large amount of my base information will be able to translate over, but I still would have hundreds of hours, if not thousands of hours of learning that would be required to translate the information that I have in my specific niche over to this other niche, just because of the nuances of those particular fields. And this kind of connects what we were discussing before. In digital pathology, I know what augmentations work, and I know when to use them and how to use them. And it may seem like a trivial thing to understand, but it took months in training of hundreds of models and reviewing the results to understand which ones actually were useful and how to use them well. So if I went to the radiology field, I basically have to undergo that process again. There's no way beyond building experience in that sort of thing. So it's not, and this kind of, I'm going to answer your question now. I think the challenge is really in combining these two. It needs to be done not as a post thought, but it needs to be done as a pre-thought from the beginning. So, for example, I will go and find the collaborator right from the onset of the project, right? I don't think of myself, I don't perform basic science. I'm not a basic scientist. I'm a translational scientist. And as a translational scientist or biomedical engineer, which is the department I'm in at Emory, our goal is to solve problems. Now, I think the most important thing that I can stress is a lot of people, in particular AI scientists or data scientists, will create problems for themselves, then solve those problems, and then go to the medical enterprise and say, I solved this problem for you. Here, take this solution. And then the medical enterprise says, yeah, but we didn't actually have this problem, or did you account for these things, or this technology doesn't fit into our workflow, or 100 other reasons why that fundamentally fails. It's a lot. As a translational scientist, my responsibility is to identify a problem in collaboration with someone else. So I'll sit down with an oncologist and a pathologist, and we'll say, what challenge is the clinician currently facing? And they'll say, we have this new drug, for example, immune therapy. We know that 50% of our patients will respond in melanoma. 50% of the patients will respond to immune therapy. 50% of them won't. We don't know currently which ones will and which ones won't. Can we build a project around this? And we say, okay, now you've outlined the problem. Pathologist, what do you think about this problem? Oh, we might be able to go and look at the way that lymphocytes are kind of interacting, because we know that immune therapy is doing this and we can see these lymphocytes on there. Andrew, can you quantify that presentation? And then I come in as a translational scientist and say, okay, so I understand that we're looking at the predictive biomarker, that we want to look at cancer cells versus lymphocytes. Can you show me some examples? Oh, I see what you're doing. Okay, for this, we can apply a UNet, and we can apply this deep learning technology, this machine learning technology, and this AI technology to precisely quantify those patterns and see if we can build a biomarker. So this is really how the system should fit together. But unfortunately, it rarely happens that way. I think mostly because the kind of more basic computer scientists, they have great ideas and they have to be doing this type of research, because we actively as translational scientists rely on it. So I read all of their papers. I read the papers from Google, even though they're not in digital pathology space, and try and understand how we can apply those technologies. But to think that you're a basic scientist and you'll discover something and immediately be able to plug it in, there's kind of a step in the middle that's often missed. And it's really this collaboration should happen from the beginning and not be an afterthought. And once you do that, then you're ensuring that you're in constant feedback. Like we have meetings with our clinicians and our pathologists every week or bi-weekly to show them progress. Oftentimes, because again, I'm not an expert in that particular disease. So their insights will say, oh, wait a second, this particular case that you're looking at is actually a rare subtype and shouldn't have been included here. maybe we should remove it. And now we have a different conversation. Or the pathologist will say, oh, this is actually a different phenomenon that's associated with this other thing. And then we go and they give us a bunch of reading, and then we read, various textbooks or whatever it is to get up to speed and realize that our assumptions were wrong and we need to fundamentally change our experiment. But we want to do this as quickly as possible in this iteration, instead of just having them give you a project, you disappear for 6 months, come back and say, hey, we spent a few thousand hours on this, look at our project. And they say, this is great, but it doesn't work for A, B, C, and D. And you say, oh, well, I didn't know those things. I'm not supposed to know those things. They're supposed to know those things. So creating the situation where we constantly get this feedback back and forth and can enrich each other, that's really the heart of this translational component. And it's unfortunately often missed by the more basic scientists.
And so I just want to quickly discuss, I guess, clinical trials, because I've seen that like there's so much AI out there, but then the AI that goes through the clinical trials and possibly gets through versus the AI that are like, I guess, the machine learning models that necessarily don't, is going to be like a massive separator, especially in the future, because we're trying to actively apply like deep learning techniques into like a hospital setting, for example. So I was wondering, clinical trials, just because they take so long, and even if they're on AI models, if we can potentially use AI to make them more efficient, and if we can, so I guess my question is, how can AI be used to potentially make clinical trials more efficient?
Yeah, so that's a really interesting question, and it happens, I think, less frequently now. So, but there's an opportunity, as you point out, for the future. The main thing that we use clinical trials for now is they provide excellent quality retrospective data. So it's not uncommon that we'll go and collaborate with a pharmaceutical company that's put out a particular drug. They have all of the data that we need, and it's of great quality because they need to do the statistical analyses and these sort of things. And then our goal is to basically build image-based biomarkers to further see if we can improve that signal, right? So they'll have usually some indication. They say, well, we gave it to this group of patients, 60% respond. That's probably already sufficient in order to get the drug approved and used clinically. But a clinician will want to know, and so will the pharmaceutical company want to know. Well, the clinician, I guess there's two different things. The clinician will want to know, is my patient in that 60%? Because if they're not in that 60%, then I want to not waste time on that particular treatment and immediately move to another treatment that's more likely to positively impact the disease progression of this particular patient. A pharmaceutical company wants to as well narrow it because it makes their product easier to sell and easier to adapt and easier to get approved. But at the same time, they're also interested in questions like, are we missing any patients? So they'll go and, for example, run a trial on a specific subtype. And if you build a biomarker that works on that subtype, the question is, can I now take that biomarker and apply it to other patients of different subtypes that clinical trial wasn't run on? Identify some potential ones that may actually respond, and then start to create secondary trials where we say, we have an algorithm or biomarker that suggests that these drugs will work on these particular patients, even though these weren't in the original scope. So now you're trying to essentially repurpose a drug that existed and is approved in one space and apply it in another space. And I think this is very exciting, specifically because and it connects with the point that we discussed before. In rarer and rarer diseases, it's hard enough to get cohorts to go and run a large-scale clinical trial of hundreds or thousands of patients. But if we can start to have algorithms that will already suggest that certain patients in these rare trials or these rare disease indications will have a positive response to these therapies, this kind of gives us opportunities to think about how we can safely deploy those drugs or develop much smaller trials or kind of prospective clinical-based studies to figure out if these patients should now be included in those that are able to receive this drug. So I think there's a very large opportunity there. We also have other opportunities in computational pathology to look at kind of the preclinical model space where, when they're trying drugs now, they generate tons of digital image or tons of blast slides that pathologists review for mice models and, pig models, et cetera, to understand if there's toxicity. And they'll review those slides and kind of say, yeah, this looks like this liver is experiencing some toxicity or whatever it may be. We have opportunities because algorithms are going to be more able to detect more nuanced signals. So maybe we can detect even pre-toxicity before it's fully evident on the slide, helping to avoid a drug going to the next stage of the trial just to kind of get found out that, oh, in fact, it was toxic. So basically eliminating drugs sooner in that process, saving all of the associated downstream costs and laborious natures. As well, there might be an opportunity to use those to identify as well, we'll say like human surrogates and say, wow, this drug is much more likely to work in humans, to better prioritize which drugs get promoted to trial without actually having to run them on humans from the onset.
And so with that being said, I want to move into the final portion of today's interview, which I want to dedicate to just, I guess, advice for the younger generation. So I'll just start off by asking, where did your initial experiences with AI come from?
So it's interesting because I guess I would say I'm old school. I existed before the existence of Google. I existed before, in fact, the existence of what we would actually call AI, before the existence of deep learning, before the existence of large language models. So I started with more routine image analysis type techniques. So I remember the first paper that came out of Andrew Ng's group from, I think it was at Stanford at that time, where they go and show that, using a bunch of CPUs, this was even before the GPUs were being used, using like thousands of CPUs, they were able to go and kind of identify cats in an automated fashion versus, dogs and these sort of things. And that was the first really, groundbreaking work that I was exposed to. And I thought it was amazing. And as soon as that came out, I realized the value of it. And I started working more and more towards that field. I was actually finishing my PhD at that time, which did not involve deep learning. It was still more traditional image analysis, handcrafted type work. But in my postdoc, I dedicated almost all of my time to deep learning and becoming familiar with it. And it really involved, I was using MATLAB at the time. There was no PyTorch. Python wasn't as popular then. At that time, I was basically shifting from MATLAB to Python, but we were writing just tons of MATLAB code ourselves. This framework called Caffe came out, which was C-based, but had like kind of a nice interface. That framework, in fact, no longer even exists, which I think is interesting, kind of like obsoleted itself. So it was really kind of an organic, slow growth with the field. And I think that was a valuable experience because it came in as a trick. And it's a lot easier to learn things when they're kind of emerging around you, because you get deep learning, 1 month, and then 3 months later you get this concept called dropout, and then 3 months later you get this concept called batch normalization. So it's really like the new information occurs at a pace that's reasonable to read the full paper, work out the math yourself, convince yourself that why this would work and how it would work, and actually internalize it, to play with it a little bit and understand those concepts before the new next concept came out. At the same time, I think there were a lot of challenges just because there was a lot of information that was, false positives. We thought we had a lot of really cool ideas, but it turns out those were just flash in the pan. They didn't really scale well. It didn't work for other data sets, or other algorithms were much more superior to those approaches. So you had the possibility of going down a lot of smaller rabbit holes. So there kind of became, as a translational scientist, we weren't really building deep learning. We were trying to apply deep learning. So for us, there was kind of like a wait and watch for a lot of these things. And we'd look at the major conferences and see what papers won, best paper that year, or which ones started to rack up a large number of citations, and try to figure out how to focus on the signal versus the noise. So for me, it was really kind of an organic growth, growing with it. We put out one of the first papers in digital pathology for deep learning in JPI, I think it was in 2016, using that Caffe network. And now it has thousands of citations. And it basically showed that we could use deep learning in a reasonable way. These GPUs were tiny back then. And we were still able to do it, right? We had far superior results than any of the state-of-the-art image analysis techniques that we and ourselves had developed and others had developed. And that kind of laid the framework and the groundwork for us to understand that, okay, this is how we need to be doing these sort of things in the future. So it really was kind of see the technology, try it out, understand that, oh, this is really nascent. It was a huge pain, right? There was thousands of hours of debugging because there were no tutorials. There was no really great framework. Like everything was really hack it together yourself type approach. But I think that really helped build the deep expertise and understand the nuance of the technology, so that now when I go to reapply it or read new papers, I have a much deeper intuition about, is this going to work on my specific use cases, or how we can reapply this sort of thing.
So with that being said, I just want to ask, I guess, the final question. And so when you were starting off in the process, I'm sure there was like a lot of obstacles, like as you were talking about just now, like the hours of debugging that you put in. So if there was something that you wish someone told you, I guess, when you started off that you think would have been beneficial, what would you like to leave, I guess, the younger generation of AI researchers, including myself, with?
So yeah, I think that's a fantastic question. I think there's maybe three parts. One is to find a mentor, right? There's no reason that you should be, trying to do all of this stuff on your own. And I think if you find a good mentor, you basically gain that experience. I mean, I think I first started programming when I was like 7 years old, and I'm in my 40s now, right? So if you could just imagine 33 years experience of programming across many different languages versus someone that's just starting out, having that knowledge about how to organize things and how to write reproducible, readable code, even the code quality itself is a huge thing in translational science, because we try and like give that around. But that mentorship is also going to help you identify problems that are worth solving to make sure that you're not wasting your time. Because I argue that with deep learning now, if you basically smash your head against the wall, eventually you'll get something that works pretty well. Now, the point is that at the end of that, especially for a young person, you want to have something that you can show the world and be proud of. And I don't think that that's going to be the same thing as like, hey, we retrained ImageNet again. Hey, we retrained ImageNet again. Hey, we retrained ImageNet again. Right? By working with collaborators, professors, or teachers, or whoever it is who have ideas for specific problems that they're facing, you can now channel that energy and channel that head smashing against the wall to a particular challenge that when you solve it, it's going to be a unique solution that you yourself can kind of take credit for, show to the world, and somehow make the world a better place. And I know that even for us, we have undergraduate students that collaborate with us. We've also worked with a number of high school students that are interested in this, and they come to us and we give them training material to get up to speed. And then we give them real projects, right? We have a lot of projects. There's a lot of interest. There's a lot of things, both large and small, that we want to do. Some things are sufficiently small that we can give it to a younger person with less experience and let them kind of mull over it. And we kind of know the answer, but don't have the time ourselves to do it. It's a great learning experience for them. Sometimes they get a paper out of it, but it promotes them to the next stage where they can then start to work on medium hard problems and then large hard problems. And I think one of the challenges is, as well, for a younger person, is to understand how to invest their time. And that's something that a mentor can help with. Because if a young person takes on a challenge that's way too hard for them, they may get discouraged. They may not be able to make progress themselves. There's some problems that I take on that are even too hard for me. And I work with other scientists and we spend hours thinking about it. And, this is really brain-racking stuff. Can you imagine if you're a young person and you accidentally take on that same challenge? Not to say that they don't have the capacity to do it, but it's going to be painful, right? There's going to be a lot more. It's going to be easier for them if there's more resources available. So I think like active mentorship and finding people to work with to make sure that your problems are good is important. The second thing I would say is to really think about what you want to do with your life and focus on a specialization, right? As I mentioned before, my specialization is in digital pathology. I know nothing about self-driving automated cars, right? I know some things about radiology. I can read the papers and understand them, but I wouldn't feel comfortable going and saying that I can go and build out a clinical system for that sort of thing. So there's a certain component that the sooner that you figure out in your life what you're interested in doing, the more that you can invest into it. And that naturally starts to build some momentum where it becomes easier and easier to do things, because now you can, for example, process a whole slide image and do an experiment in a day instead of a month. And if you keep jumping around from different fields, which you should do, right? You should do to figure out what you want to do, but you shouldn't do it haphazardly. You should constantly be thinking about, how can I find a particular niche that I'm really interested in, that I'm willing to commit to learning all of the nuances of? Because that's really where the next generation of science is going to take place. It's not going to come from general data scientists that just run around with a hammer saying, look, I can go and hit these nails. It's really going to become understanding very deeply a problem and coming up with custom fit solutions for that problem in line with all of the different stakeholders that are invested in that particular space. And I think the last thing is what I find is most students will tend to focus more on looking at summary statistics and looking at the results at a higher level of their classifier, and they're afraid to go back or to their original data because maybe there's so much data or whatever it is. I think the challenge is, especially in the medical space, we're never going to get 100% accuracy, right? So 100% accuracy is out of the question, especially for predicting which patients are going to respond to a particular drug. We just don't have that technology. I don't think 100% of that signal is embedded in a deep learning image as well, or a digital pathology image as well. So then the question becomes, what are the false positives? Why are those false positives? What are the false negatives? Why are those false negatives? And then once we start to understand this goes and allows us to better understand how to improve that system. So for example, when we discussed augmentation before, what I would typically do is I would train a model. I have a model. Instead of applying that model to the validation set, I'll apply that model back to the training set. And I'll go and look at where examples, right? Maybe it's 100 images. I'll look at 100 random patches that were incorrectly classified and start looking at them and saying, what's common about these? What is there something here that I'm missing? And then when you start to do that, you realize, oh, all of these images are, for example, much brighter than the other images in the data set. Maybe I should do some type of brightness augmentation or brightness normalization to improve the performance. And then that becomes the next step of the classifier. We spend a lot more time doing quality control of the data and review of the data than I think most people expect. So one of the challenges is that when you, for example, look at challenge data that you can find on the internet, that data tends to be very, very heavily curated. And when people take deep learning courses in school, that data was also very heavily curated, because the goal of that is to learn how to train a classifier and to make sure that it converges and to learn all these different kind of properties of it and to get familiar with that. But that data is not the same type of experience that you have when you walk into the real world. Real world clinical data is very different, and it's very messy. And I can tell you an anecdotal story of a student of mine that came in, and his intuition, which is, I understand why he had this intuition, was he was trained as a data scientist. He had a master's degree in data science. Was to go and, like, his results were 70% accurate, we'll say, from the beginning. And he said, well, I'm going to go and try a different model. I'm going to try a different model architecture. I'm going to try a different model architecture. And he wanted to keep doing this for a number of weeks. And I said, listen, this model architecture is not going to fundamentally change your performance. You need to look at the actual data, and we'll figure it out from there. Eventually, he was, kind of reluctant to do this. And I said, why don't you try it your way for 2 weeks, and then you try it my way for 2 weeks. So for 2 weeks, he tried, maybe 10 or 15 different architectures, different model sizes, vision transformers, CNNs, all of this sort of stuff. And over that time period, he saw maybe a 3% performance improvement in that accuracy measure. I was like, now we're going to do it live. And we start going and just reviewing the actual input data itself. And he's like, oh, but these images are blurry. I was like, should you be training on blurry images? He's like, oh, probably not. I was like, so he's like, oh, I'm going to do some quality control. Oh, these were actually incorrect inputs. These are false positives. These were mislabeled. These were positives, but were actually negatives. Oh, this is a specific case of this. Then he spoke with the pathologist. They said, oh, no, you should actually do it like that, blah, blah, blah. Over the course of that time, right, now he spent the rest of his master's degree working on this. 80% of it was quality control and improving not the model itself, but improving the data, how the data was represented and the quality of that data. And then by the time he finished, he had 90% accuracy on that. So he improved 20% by using literally the same deep learning code that he used on the first day. Did not change a single piece of that, the actual model, the number of parameters, all of that sort of stuff. Instead, he changed augmentations and he changed the label of the data. He filtered the data, and then that's how he got it down. And this ties with this overarching point that I'm trying to make. You need the expertise in order to be able to figure out how to filter and perform that type of adjustments. It's not changing the model that's going to get you 15% extra performance. It's really deeply understanding your domain. And that takes a lot of time, a lot of years, a lot of collaboration, a lot of discussion. And it's an investment that's absolutely worth it. I hope from my example that I just showed you that once you have that knowledge, you can employ that knowledge well and get 25, 30% improvement. But it takes time to gain that knowledge. But the next time he goes to apply that knowledge to the next data set, it took him 1 week to do it. Why? Because he said, oh, I'm just going to review the data. Oh, no. I should change this augmentation. I should do this sort of stuff. And then he personally got faster. So I think it's about committing or finding a field that you're interested in. Of course, try a number until you you're happy with what you found. But the sooner that you commit, the sooner you build that momentum, the sooner you can start taking on harder and harder problems. But a basis of this is really understanding the domain, not getting distracted by summary level statistics, and really going and looking at the individual data points that you have, performing that quality control, performing that data review. And if you do that well, then the summary statistics will naturally improve as a result.
Well, that's all for today, and thank you so much for joining me here.
Yeah, thank you. It's been a pleasure.
For anyone watching this, make sure to leave a like, subscribe, and comment down below.